I'll take a few minutes to talk a little bit about Carisma and what we're working on, and then I think Bola has a few questions. Carisma is a relatively new company. We're about 3 years old. We're working on a platform called CAR macrophages. This is some technology that was licensed from Penn and co-founded by Sar Gill and Mike Klaczynski. The work is actually Mike Klaczynski's PhD thesis and under advisement by Sar Gill and Carl June. We believe that CAR macrophages are ideally suited cell therapy approach to the treatment of solid tumors. Not the least of which reasons are that there's a dual mechanism of action, both phagocytosis and antigen presentation. So effectively what we're doing is we are generating or mimicking an innate immune response to generate an adaptive immune response. We have an emerging pipeline, one product under development that should enter the clinic hopefully by the end of this year. Our target is to have an IND filing in Q4 and enter clinic as soon as possible after that. We have additional programs that are under development. We have 2 other constructs that we're making against other antigen targets, and once we have them back in hand, we'll say what those antigen targets are. We're well financed. We raised $59 million last year in a Series A. Our investors is a strong syndicate of strategics as well as traditional investors. The deal was led by AbbVie and HealthCap. In addition, we brought in Wellington Partners, IP Group, TPG Biotech, Agent Capital, and Merck or MRL Ventures. We have a great team. We're a growing company. I was prepared to say 14 employees this morning, but we just brought another person on. So we actually have 15 employees now as of this moment, and a great team of advisors and a traditional, as you might expect, venture-backed board. Our technology can be summarized on this page, and essentially what we do is we take a macrophage, harvest it from a patient, or we harvest monocytes, differentiate into macrophage, and insert a CAR. We would re-administer to the patient, and through a chemokine gradient, the macrophage is trafficked to the tumor. They'll engage the target, and as you'll see, they will devour the target. Whoops, sorry. I guess there isn't a pointer. They'll eat the target and killing that, and then process and present neoepitopes from within that cancer cell. Then, using the macrophage's professional antigen presenting capability, they will present those neoepitopes out and generate an adaptive immune response. So we have generated data along the way, and unfortunately, given time constraints, we're not able to go into that. But we are really excited about the technology and think that it is a potential game changer as an approach for solid tumor treatment. In terms of our highlights and what we've accomplished so far, in 2018, we completed our financing. We initiated our manufacturing. That is obviously the long lead item for our IND. We actually started that late in 2017. We've partnered with a couple of different area experts, Oxford Genetics for our plasmid, WuXi AppTec for our viral vector, and Miltenyi for our cell product manufacturing. I mentioned the Series A. We closed $59 million last year. We've established our own company infrastructure. We have lab facilities adjacent to Penn at the Biolabs facility in Philadelphia. We can basically do everything that we were able to do initially at Penn. We can do it at our own site. And then finally, towards the end of the year, in December, we did lock down a key element of our manufacturing process, which is our viral vector manufacturing. As we look forward to 2019, for the rest of the year, there are a number of things that we're trying to do. One, round out our management team. So I am recruiting a CSO, CMO, and CFO to join the team, and that will give a full management team across the board. We'll continue to grow the research and development staff and anticipate ending the year with around 30 FTEs. We hope to finalize our cGMP manufacturing process by late Q3, early Q4, and then that will lead to an IND filing and initiation of our phase 1 study. Our phase 1 study, by the way, will be targeting HER2. So we have a HER2 CAR macrophage. That is our lead program, and it'll be a traditional basket design where all comers, and we'll be looking at measuring safety, obviously, but early indicators of efficacy, including trafficking to the site of the tumor, phagocytosis, antigen processing and presentation, and an adaptive immune response. And then lastly, this is an early stage program, first generation approach, and Mike Klaczynski, one of the co-founders, has joined the team as VP of Discovery and has a wealth of ideas on how we can improve the platform in subsequent generations, including things like adding in macrophage specific signaling domains, non-viral gene delivery potential, looking at other cell types to use for our approach, like, for example, looking at monocytes or even earlier upstream. So that is us in a very short presentation. If people are interested, we certainly can reach out and ask us about other things. We have a wealth of data supporting the direction that we're taking.
Great, thanks. So let's start by chatting about your lead program. It targets HER2 positive cancers. Can you talk a little bit more about why that was a good starting point for your technology? It should work without pushing anything, I think.
Right. I don't think it does work.
Does it work? Oh, we can pass the mic.
Great. So yes, that's one of the early discussions as we were looking at where to go, and so there are a number of solid tumor antigen targets that people have pursued. HER2 was selected for a number of reasons. Number one, it was a well understood antigen target with prior therapy, Herceptin obviously, and then other anti-HER2 approaches. We also need a cell surface antigen target, and even upon resistance, the antigen target is present. We had seen, aside from one instance of toxicity associated with an engineered cell against HER2, fairly clean safety profile with engineered cells, with CAR-T approaches. And lastly, the antigen density on other tissues is relatively low, and we believe that we had a good therapeutic index with the antigen density you'd see on tumor cells.
Great, and so the technology CAR macrophages, is that a cancer technology only, or do you plan to go into other applications?
If you think about the technology itself, macrophages are the body's Pac-Man. They just patrol and eat things that shouldn't be there. What we're able to do is target them against specific cell types and specific proteins. While it lends itself well to tumor cells, you could think about any other protein or cell target that you could direct a macrophage against. So we have considered things in cardiovascular space, in the CNS space, fibrosis, a number of other areas. We currently don't have the bandwidth to pursue those, but it's something that we have considered and would explore through a partnership.
Great. And so back to cancer, CAR-Ts, in terms of applications in cancer, sometimes are thought of as one-time treatments. Would that be the case for CAR macrophages as well?
No. So I think there's a couple of different facets here. If phagocytosis were the only mechanism that works, then I think repeat administration of macrophages would work as a debulking or a repeat treatment. What we're hoping, though, is to stimulate an adaptive immune response. In the event that we are successful in doing that, then it probably would translate into a one-time treatment, but it's certainly not precluded from doing multiple doses.
Sure. And we know that manufacturing has been an important topic for CAR-Ts. Can you talk about your process and compare and contrast, basically? Is it as challenging?
Sure. I think that we learned a lot observing what was happening with CAR-T manufacturing. And the basic process is the same. We harvest cells from patients. We end up differentiating to macrophages. So we harvest CD14 positive monocytes, differentiate into macrophages, introduce the CAR construct through a viral vector. In our case, it's a Ad5F35 adenovirus chimera. It turned out that, as Mike was going through the work, that neither lenti CAR-T nor AAV would work in terms of transducing macrophages. Our Ad5F35 has about a 75% transduction efficiency. And so we introduced the vector, or the viral vector, and are able to generate a CAR. We freeze at 7 days. So the difference is, so in general process, it's about the same. The time period is about a third what you would see, but essentially it's the same. And I think that some of the engineering work that's been done on the CAR-T side is helping us as we progress.
Great. I want to go back to sort of understanding the technology. So we know T cells are great for engineering. Could you talk a bit more about why macrophages would be great to engineer as well? And might you be able to go after a different set of indications relative to the ones appropriate for T cell engineering?
Sure. I think in the inception of the company, the idea was we're looking at CAR-T and the challenges that, while obviously great outcomes in hematologic malignancy, the challenges in solid tumors were multifold. We believe you can boil it down into three separate areas. First, trafficking. Access to the tumors by T cells is fairly challenging. Macrophages, on the other hand, are actively recruited to the site of the tumor. And in fact, if you look at the cell types in the tumor microenvironment, the number two cell there is a macrophage. So we know that they can actively transport. Secondly, it's immunosuppressive microenvironment, and so whereas T cells are again challenged there, our activity is really separate from that. So we can go in and we're not really affected by that environment, and we can phagocytose the target of choice. In fact, we believe that we can release cytokines that may make it a more conducive environment, recruiting T cells and NK cells. In vitro, we've been able to show conversion of some M0s over to M1. We may be able to do the same with TAMs, and so it just remains to be seen. So those two are important aspects. The last would be looking at a CAR-T approach. Would be there's issues with tumor heterogeneity or antigen heterogeneity between tumor cells. Here, what we're looking at is targeting a ubiquitous antigen that is only there for targeting purposes at the outset. What we're looking for is neoepitopes from the cancer cells and generating, again, a true adaptive immune response. And so the loss of antigen shouldn't be a problem for us.
Great. And so much of oncology these days, it's about combinations. So are there any particular regimens that in theory might be logical for your approach? What else might work with CAR macrophages in theory?
Yeah, so I think that it's early days to say. I think the first step we need to do is to demonstrate safety. While there's interest in other approaches for macrophages, like the CD47 SIRP-alpha pathway or repolarization, those may lend themselves to working with us. So that's certainly an area we'd want to explore. Other means to warm up a tumor, looking at checkpoint inhibitors, may also lend themselves to a CAR macrophage approach. But obviously, single agent efficacy is really what we're looking for at the outset.
Right. And you named some pretty interesting human capital earlier on. So you have access to some giants in the industry. Are there any particular advantages that you have conferred to you from the collaboration, or at least the association with Penn or any of the, let's say, scientists you work with?
Yeah, so I think that it's great being able to work with SAR and Carl as advisors as we're going along. It certainly helped Mike as he was proceeding. I think that they're both actively involved in giving us advice and helping guide the company. We also have good relationships, as you know, with the capital that we raised in some strategic partners or advisors, I should say, not partners, because it's through their venture arms. But both AbbVie and Merck have certainly given us access and resources that we may not see as a tiny company alone.
Great. And final question, hidden value, or what investors are missing. What is it that you have to explain that isn't necessarily apparent? What would you like investors to know about the company?
Yeah, I think that in its most simple form, and I always equate it to, we're engineering a cell to an innate immune cell, and what we're really trying to do is drive true adaptive immunity. And I think that's a differentiating characteristic for what we're doing relative to other cell engineering plays.
Great. Thank you, Stephen.
All right.